Hello everyone, it's Jihad Ali from Grand Canyon University, and the course is Concept of Human Physiology, and today we are going to talk about the muscular system. So without further ado, let's get started. So what are the structures of skeletal muscles? Skeletal muscle fibers contain numerous myofibrils, which are cylindrical intracellular structures 1 millimeter in diameter that extend the entire length of the muscle fiber. Myofibrils are specialized contractile elements that constitute 80% of the volume of the muscle fiber. Each myofibrils consists of regular arrangement of highly organized cytoskeletal microfilament, which are the thick and thin filament. The thick filament, which are 12 to 18 nanometer in diameter and about 1 to 0.6 millimeter in length, are special assemblies of the protein myosin, whereas the thin filament, which are 5 to 8 nanometer in diameter and about 1 millimeter long, are made up primarily of the protein actin. Myofibrils display alternating dark bands, the A bands, and light bands, the I bands. The bands of all myofibrils line up parallel to one another, collectively produce the striated appearance of skeletal muscle fibers visible under a light microscope. Alternate stacked set of thick filament that slightly overlap one another are responsible for the A and A band. What is A band? A band is made up of stacked set of thick filament along with the portion of the thin filament that overlap on both ends of the thick filament. The thick filament lie only with the A band and extend its entire width. The light area within the middle of the A band, where the thin filament do not reach, is called the H zone. Only the central portion of the thick filament are found in this region. Supporting proteins that hold the thick filament together vertically with each stack can be seen as the M line, which extends vertically down the middle of the A band within the center of the S zone. I band, on the other hand, consists of the remaining portion of the thin filament that do not project into the A band. Visible in the middle of each A band is a dense vertical Z line, which is the area between two Z lines is called sarcomere, which is the functional unit of skeletal muscles. A functional unit of any organ is the smallest component that can perform all function of that organ. Sarcomere is the smallest component of the muscle fibers that can contract. The Z line is a flat cytoskeletal disc that connects the thin filament of two adjoining sarcomeres. Each relaxed sarcomere is about 2 millimeter in width and consists of one whole A band and half of each of the two I bands located on either side. I band contain only thin filament from two adjacent sarcomeres, but not the entire length of this filament. During growth, a muscle increases in length by adding new sarcomere on the ends of the myofibrils, not by increasing the size of each sarcomere. Single strand of giant, highly elastic protein known as titin extend in both directions from the M line along the length of the thick filament to the Z line at the opposite end of the sarcomere. Titin is the largest protein in the body, made up of nearly 30,000 amino acids. What is a cross bridge? Cross bridge under electron microscope can be seen extending from each thick filament toward the surrounding thin filament in the area where the thick and thin filament overlap. In this slide, we are going to talk about the molecular basis of skeletal muscle contraction. The thin filament on each side of sarcomeres slide onward of the stationary thick filament toward the A band centers during contraction. The thin filament pull the Z lines to which they are attached closer together so that sarcomeres shortens. As all sarcomeres throughout the muscle's fiber's length shorten simultaneously, the entire fiber shortens. This is the sliding filament mechanism of muscle contractions. The H zone is the center of the A band where the thin filament do not reach, become smaller as the thin filament approach each other when they slide more deeply inward. The I band, which consists of the portion of the thin filament that do not overlap with thick filament, narrow as the thin filament further overlap the thick filament during their onward slide. The thin filament themselves do not change length during muscle fiber shortening. The width of the A band remains unchanged during contractions because its width is determined by the length of the thick filament, and the thick filament do not change the length during the shortening process. Note that neither the thick nor the thin filament decrease in length to shorten the sarcomers. Instead, contraction is accomplished by thin filament from the opposite side of each sarcomer sliding closer together between the thick filaments. Let's talk about the bow stroke. The bow stroke during contraction with the tubomycins and troponin pull out of the way by calcium. The myosin head or cross bridges from thick filament combine with the actin molecules in the surrounding thin filament, myosin. Myosin cross bridges walk along an actin filament to pull it inward relative to the stationary thick filament. The two myosin heads of each myosin molecules act independently, with only one head attaching to actin at a given time. When the binding sign on actin molecules is exposed, the myosin molecules tilted at the hang point on the tail, elevating the myosin head to facilitate the binding of these cross bridges to the nearest actin molecules. On binding, the myosin head tilts 45 degrees inward, bending at this neck hinge point, create a shocking motion that pulls the thin filament toward the center of the sarcomers. The action is known as the bow stroke of a cross bridge. A single bow stroke pulls the thin filament onward only a small percentage of the total shortening distance. Repeated cycle of cross bridges binding and bending complete the shortening. At the end of one cross bridge cycle, the length between the myosin cross bridges and the actin molecules break. The cross bridge returns to its original angle and bind to an actin molecules behind its previous actin partner. The cross bridge tilts onward again to pull the actin filament and further, then detach and repeat the cycle. Repeated cycles of cross bridge bow stroke successfully pull in the thin filament. Because of the way myosin molecules are oriented within thick filament, all cross bridges stroke toward the center of the sarcomer, so that all 6 of the surrounding thin filament on each end of the sarcomers are pulled inward simultaneously. The cross bridge aligned with the given thin filament do not all stroke in union. However, at any time during contraction, part of the cross bridges are attached to the thin filament and are stroking, while others are returned to their original conformation in preparation for binding with another actin molecules. Skeletal muscle fibers contract and relax at different speeds. They vary in structure and function in which metabolic reaction they use to generate ATP, and also on how quickly they fatigue. For example, a fiber categorized as either slow or fast twitch depending on how rapidly the ATPs in its myosin head hydrolyze ATP. There are 3 different skeletal muscles fibers known as type 1, type 2A, and type 2X fiber. The difference of size, color, and contraction speed and contraction force and energy source classify each fiber. For example, type 1 fiber, also known as slow twitch fiber, are the smallest fiber types with darkish red color. It has a fairly slow twitch speed and produces a relatively small amount of force when contracted. It has the high amount of mitochondria, which are organelles, as we know, within each cells that use oxygen to produce energy. Although the force generated is fairly small, type 1 fibers are highly fatigue resistant, allowing it to be active for a long period of time. They are the primarily fibers using low intensity activity with steady oxygen consumption, such as walking, jogging, or aerobics. Type 2A fibers, on the other hand, aka moderate fast twitch fiber, also known red but intermediate in size. These larger fibers typically use a combination of oxygen and glucose as source of energy. This combination allows for quicker contracting speed and higher force output compared to type 1 fibers. However, fatigue resistance isn't as high as type 1 fiber. These fibers are typically activated during anaerobic activity that are moderate in duration, such as mile runs, swimming, and short distance cycling. Type 2X fibers, which is known as fast twitch fibers, are white in color due to a low oxygen capacity and by far the largest fiber type. It makes up for the lack of oxidative capacity by having extremely high level of glucose in stored form of glycogen, producing the fastest twitch speed and the most force contraction. The downside of these fibers is that the fiber fatigue is quickly burning out after 15 to 30 seconds. So high impact, heavy resistance activities such as lifting weights and serpentines will activate type 2X fibers the most. For any activities, your muscles follow certain recruiting order that slotless, low force, fatigue resistant type fibers are always activated first. When type 1 fibers are maxed out, type 2 fibers are activated, and then after those are maxed out, then type 2X fibers are then activated. This order is known as Henneman's size principles, help minimize physical fatigue and allow precise motor control by using no more than force necessary to complete movement. Everyone has a genetically determined amount of each muscle fibers type. Some people are born with a predominant amount of certain muscle fibers, making them effective with activity that favor those fibers. There have been more findings that suggested that type 2X fibers can change into type 2A fibers with proper training. However, this might simply be due to type 2X fibers showing higher oxygen capacity through physical adaptation. They are ultimately still type 2X fiber. The length tension relationship of skeletal muscle cells describe the force of contractile at different muscle length. We recall that the sarcomere is the function unit of muscles where contractions occur. When a contractions begin, myosin must bind to actin to form cross-bridges with each other, resulting in a power stroke. Several of these interactions will occur in a cycle so that the two ends of sarcomeres come closer together. Depending on the number of cross-bridges formed will determine the force of contraction. If the sarcomere is hyperextended, there are less cross-bridges, which means decreased tension and ultimate weak force of contraction. If there is a muscle is hypercontracted, there are too many cross-bridges formation, so the tension is so high that there is little or no movement, also resulting in low force. The sweet spot is somewhere in the middle, where the sarcomere has an equal number of overlap between the actin and myosin. Length tension curves show the relationship between the length of one sarcomere and the tension that can be generated. So let's talk about the active tension. So the active tension is in the length tension curve is generated by the contractile element of sarcomere, that is the traction between the myofibrils. The peak tension is produced when the sarcomeres are at their lasting length. At this moment, provide the optimum alignment between the actin and myosin filament. When the length of sarcomeres shorten, the actin filaments are pulled along the myosin filament, which in turn pull the Z lines closer to the myosin filament. At extremely shortened length, T10 is compressed between the myosin and the Z lines, and the actin filament overlap each other centrally. A consequence of extreme shortening is that there is no further potential for myofibrils to contract, and there is no force can be generated. During sarcomere lengthening, actin move in the opposite direction along the myosin. At extreme length, the myosin binding site on the actin filament move out of the range of the globular heads of the myosin filament. A fewer myosin heads combined with actin filaments, lower force is generated, form myofibril contractions. So what's passive tension in the length tension curve is generated by the barrel and series elements, and therefore doesn't contribute to the generation of the tension with shortened length. Technically, the passive line should be at zero force for the length shorter than resting length. though typically the line is absent in the region of the length tension curves. As sarcomeres length increase, the passive tissue reach their full length and start to provide resistance to further increase in length. So we should know that the ratio of the contribution of the passive tissue to the total tension increase until the maximum length are reached. At the maximum length, tension or resistance to an increase in length is virtually all passive. Molecular basis of smooth muscle contractions. Smooth muscle cells are spindle-shaped, tapered at both ends, have a single nucleus, and are considerably smaller, 2 to 9 nanometer in diameter and about 50 to 400 nanometer long. Also, unlike skeletal muscle cells, a single small muscle cell doesn't extend the full length of muscles. Instead, a group of smooth muscles are typically arranged in sheets. A smooth muscle cell has three types of filament: thick myosin filament, which are longer than those in skeletal muscles; the thin filament, which contain topomyosin but lack troponin; and the filament of intermediate size, which doesn't directly participate in contraction but are part of the cytoskeletal framework that support the cell shape. Smooth muscle filament do not form myoprofiles and are not arranged in the sarcomeres pattern found in skeletal muscles. Thus, smooth muscle cells do not show the banding or striations of skeletal muscles. Hence, the term smooth for this muscle type come from. Lacking sarcomere, smooth muscles doesn't have Z-lines, but it does have dense body containing the same protein constitute found in Z-line. Dense bodies are positioned throughout the smooth muscles, as well as attached to the internal surface of the plasma membrane. Dense bodies are held in place by a scaffold of intermediate filament. The active filaments are anchored to the dense bodies. Considerably more actin is present in smooth muscle cells than in skeletal muscle cells, with 10 to 15 thin filaments for each thick myosin filament in smooth muscles compared to the 2 thin filament for each thick filament in skeletal muscles. The thick and thin filament contractile units are oriented slightly diagonally from side to side, with the smooth muscle cells in an elongated diamond-shaped lattice, rather than running parallel with the long axis as myoprofiles do in skeletal muscles. Relative sliding of thin filament past the thick filament during contraction causes the filament lattice to shorten and expand from side to side. As a result, the whole cell shortens and bulges out between the points where the thin filaments are attached to the inner surface of the plasma membrane. Unlike in skeletal muscles, myosin molecules are arranged in smooth muscle thick filaments so that the crossover bridges are present along the entire filament length. That is, there is no bare portion in the center of the smooth muscle thick filament. As a result, the surrounding thin filament can be pulled along the thick filament for longer distance than in skeletal muscles. Also, dissimilar to the skeletal muscles, in which all thin filaments surrounding thick filaments are pulled toward the center of the stationary thick filament. The myosin protein in smooth muscle thick filaments are organized so that half of the surrounding thin filaments are pulled toward one end of the stationary thick filament and the other half are pulled toward the opposite end. Thank you for listening, and here are some references.